Good morning. Thank you all for coming. My name's Ed Tate. I'm with the Office of Research Alliances, Rutgers. And we're the organizer of this event. The co-sponsors are Sigma-Aldrich, Supelco, and Elsevier. And thank you all for coming. Our next speaker is Joe Frangipane with Sigma-Aldrich.
Thank you, Ed. So I'm a field application specialist for the East Coast covering functional genomics products with Sigma-Aldrich, and today I'll be going into specifically functional genomics products geared toward knocking out and knocking in new genetic elements into various genomes. So just by way of introduction, the things that we'll cover today will be just some basic information on zinc finger nuclease technology, what it is, how it works, how it was constructed. Some examples of targeted gene knockout and also targeted gene integration, so some real knock-in and knockout data in cells, cell lines, and also in stem cells. And then going into a little bit about the product line itself and what Sigma offers around both custom and off-the-shelf zinc finger nucleases. Then I'll go into some brief information about the creation of transgenic animals using ZFNs and how Sigma has made some great strides in providing both catalog and custom animals. And then finally, a description of that service. So what is a zinc finger nuclease? Basically, a zinc finger nuclease is a reagent that creates a very specific double-stranded break in just one exact gene in the genome of choice. And this is a very tall order. Obviously, regular endonucleases will cut pretty much all over the place in the genome. So it's specific, but it's a frequent cutter, so to speak. So you want to find something that's just going to bind in one spot and just knock out and just cut in that one genetic locus. So the originators of the technology, which were a company called Sangamo, and they've licensed the technology to Sigma for research. The pioneers of the technology looked to a binding motif that would allow this level of specificity. And they found it in zinc finger proteins, which is the most abundant DNA binding motif in nature. It's conserved across all different species of eukaryotes. And not only that, but it's it has a lot of different types of binding motifs. So it has versatility in that every different zinc finger protein will bind 3 different letters of DNA. So these codons can then be mixed and matched to create longer arms of specificity so that you can bind in just one spot in the genome and achieve that single double-stranded break. Here's an example of a 3-finger zinc finger protein which is bound to 9 letters altogether of DNA, so 3 each. And you can see that as we add more and more of these zinc finger proteins, we can create longer and longer stretches for targeting different genes. The other thing is zinc finger proteins are very modular, but they do affect each other's binding when they come together as doublets because of conformational changes. So all of that binding differences when 2 modules get together has also been characterized and entered into the algorithm that we use to generate zinc finger proteins and zinc finger nucleases. So all of those different interactions are part of our database for when we run design. And you can see that with this knowledge we can build 12, 15, and 18 bases of specificity. And now we're getting to that statistical probability that this is going to cut in just one spot in one gene of choice. So that takes care of the binding motif. What about the catalytic domain? Well, the originators of the technology looked at this FOLK1 endonuclease because it's modular in structure. And as a result, you can cleave off the native DNA binding motif and add your own, which in this case would be the zinc finger proteins that we choose to design, and hook it up to the nuclease element of the FOLK1. So the idea behind this is the prototype of a mature ZFN, of the binding domain as well as the catalytic domain. And this is, again, the prototype of the ZFN, but the mature ZFN took it one step further, which is to break the molecule up into 2 different subunits. So now we have a 5' and a 3' molecule, each one with its own specificity to one area of this gene locus. And now the catalytic domain is not just FOLK1, but it's a heterodimer of a single FOLK1. And this heterodimer will not function unless it comes to within 5 to 7 bases of each other. And in that way, it can function as a complete molecule and make that double-stranded cut. That 5 to 7 bases is very stringent, so this allows for very, very exact targeting and also minimizing off-target effects virtually to nothing. So this is the mature offering that Sigma offers as far as ZFN technology. So that covers what it looks like. What does it actually do to cause this knockout? Well, targeted disruption is caused by non-homologous end joining. The idea behind this is you have your double-stranded break, and you can either have an error-free repair, which you see up here, Or some exonuclease activity can chew back, and then during non-homologous end joining, which is a non-templated repair mechanism, you can get small insertions and deletions of bases. And if these are out of frame, basically the gene will then suffer either nonsense-mediated decay or premature termination. And in either of these cases now, the gene will not be transcribed successfully. You won't get a functional protein. So the gene is in effect knocked out because of this frame shift. And it's all mediated by the double-stranded break and then the non-homologous end joining. So now I'd like to go into some examples of targeted gene knockout. This is an example that was done in CHO cells, and we were knocking out the dihydrofolate reductase gene in CHO cells. So ZFNs were designed in silico with our algorithm. They were all built out, and then they were validated in CHO cells to find the most efficient cutter. And then that was the one ZFN that was used for the experiment. These ZFNs were then transfected into pools of cells, and the pools of cells were then looked at with a PCR mismatch assay to look for genetic hits where we see that there's some sort of mutation going on. I'll go into some detail about that PCR mismatch assay a little bit later in the talk. And once we found some hits in those pools, then we dilution clone those out and find the actual clones that had the genetic changes and sequence those to find out what's going on at the genetic level. So at the genotypic level, you can see here this is the wild type sequence, and this is the area to which the ZFNs bind. And then looking at these different clones that were isolated, you can see small insertions or slightly larger deletions that cause this frame shift effect that I mentioned. And this experiment was done a couple of different ways, with and without serum, and the results were averaged together. And this is typical of a ZFN experiment in cell culture. So these were biallelic mutants, so both alleles had these mutations. You can also get monoallelic mutants at about a 2x frequency. And in general, the efficiency of ZFNs, depending on the cell that you're looking at, the cell type, can average somewhere between 1 and 20%. So basically 1 in 100 clones or 20 in 100 clones. It's a very nice high efficiency for this type of experiment. And as I mentioned, in one go, in one experiment, you can actually knock out both alleles. At the protein level, we also wanted to confirm that this gene was, in effect, not coding for protein. So you can see by Western blot, the dihydrofolate reductase gene in our knockouts here is not showing any presence of the protein on a Western as compared to wild type. And so we also wanted to look a little bit deeper than that and not only confirm the protein was gone, but also that the activity was gone. So looking at that, DHFR is responsible for folate metabolism. So looking at cells that suffer from this defect, they're only going to be growing efficiently and successfully if we supplement with an HT medium. So in the presence of the HT medium, you can see that all the cells are happy and growing very well. In the absence of that, only the cells that are wild type will grow. So the ones that have the knockout, the clones here that have the knockout of DHFR, cannot grow without the rescue medium. So this shows a very, very good phenotypic change in the cells that we chose to edit with the ZFN. Now I'll go into some different types of targeted gene integration. This is the other side of the coin for ZFN. So not only can we do knockouts, but we use not NHEJ, but we use homologous recombination to get knock-ins. The idea here is to transfect our ZFN constructs, the 5' and 3' molecules, along with a donor plasmid. And the donor plasmid contains homology arms to the ZFN cut site, and including an inserted piece of DNA that can be up to about 10 kb. And using homologous recombination and the double-stranded cut, you can get a repair which involves insertion of this element into the genome, into the chromosome. And the nice thing about this is homologous recombination is ramped up and about 1,000 times more efficient in cells that undergo a double-stranded break. So rather than having to resort to doing homologous recombination with just stem cells, you can now use the ZFN to get this sort of targeted insertion in pretty much any cell type that you want to try. So this frees us up to do a lot of things, especially with embryos of animals. So I'll go into some different approaches to targeted integration using stem cells now. These are all experiments with stem cells where we either interrupted an exon with another genetic element, or we did insertion into a safe harbor locus, or we created a new exon in an intronic space of an existing gene. So in the first example, we were taking the exon for CCR5, the gene for CCR5, in human embryonic stem cells, and interrupting it with an insert, which was a GFP gene, which is under the control of a PGK promoter. So in this way, we're kind of doing a knockout, knock-in. We're interrupting CCR5, and we're selecting for cells that have positive GFP. We wanted to look at this pre- and post-differentiation in these stem cells. So you can see this was a 5% targeted insertion event. So this is, again, very, very high efficiency. And looking at GFP in the pre-neuronal cells here before differentiation, you can see that GFP comes up nicely. Also comes up post-neuronal differentiation. And this is just a marker for OCT3 and OCT4 promoter, so a measure of stemness in the stem cells. And this is a tubulin, a neuronal tubulin gene, so a measure of differentiation, to show that we can integrate, express GFP, and maintain it post-differentiation. Now I'd like to look at some safe harbor insertion. So human genome has one safe harbor site that is basically an AAVS1 site. It's a landing pattern or landing area for adeno-associated virus. And when the virus gets into that particular site, it doesn't perturb, as far as we know, anything else in the genome. So it's considered a true safe harbor. We have similar types of sites within the genomes of rat and mouse, which are known as ROSA 26 loci. And with these loci, you can insert genetic elements in there. So what we did was we capitalized on that particular type of safe harbor with a pair of ZFNs that are designed to cut at AAVS1. And when that cut is made, we also have a donor plasmid. That donor plasmid has a polylinker which allows you to clone in your gene of interest. And then using these homology arms to the AAVS1 site, you can get targeted integration of your gene of choice using the ZFNs in a very high efficient manner. So the benefits here are you're not going to get targeted insertion anywhere else in the genome. So it's definitely directed, it's not random. It's going to be at one level of expression from one site rather than multiple random expressions all over the genome. So the idea here, this is a proof of concept experiment, and this was done by Hockemeyer in Nature Biotechnology. This is the construct that we use to do the insertion. It has 5' and 3' homology arms, puromycin resistance, and also a TET-inducible GFP. Just again to see if we could get this into safe harbor and look at expression of GFP. So this was done as a monoallelic insertion and also a biallelic insertion with the TET-inducible GFP in the absence and the presence of doxycycline. And you can see that once we induce with dox, we've got GFP coming up nicely both before subcloning and after subcloning. And this shows that we can do this in a reliable way with high efficiency in one or both alleles. So the final application is splice acceptor fusion into an intron, creating a new exon basically. And in this way, what we wanted to do, again, was a proof of concept with GFP. But in this time, this is in stem cells under the control of the OCT4 promoter. The OCT4 promoter, again, is a measure of stemness. So as long as these are stem cells, you should get expression of GFP within this new exon. And then when we differentiate, this should go away. So looking at this under Western blot, you can see this effect. So before differentiation, these embryonic stem cells have OCT4 expression and also GFP. And then once they're differentiated into fibroblasts, you can see the OCT4 is going away, and the GFP expression also goes away. So this shows the ability to create these new inserted pieces of DNA, these new genetic elements, as new exons. And we did it on either side of the intron with 2 different ZFN pairs. Also, these modified cells will retain the capacity to differentiate into a wide variety of tissue, as good stem cells should. So the advantages of ZFN-mediated editing is, there are many actually, rapid permanent targeted editing in an endogenous locus or into a safe harbor locus. You can do this in many different types of cells, somatic cells, cell lines, stem cells, even embryos, which allows you to do a lot with animal creation. You can do this in monoallelic, biallelic, or with polyploid cells. You can do this in all the different alleles. If it's, let's say, 4 alleles, you would probably do 2 or 3 rounds of ZFN experimentation to get that all the different alleles to be knocked out. But if it's just 2 alleles, you can get that in one go. And minimal screening effort is required because of the efficiencies here. You only need to screen a few hundred clones. You don't need to do selection if you don't want to. In some cases where we're doing stem cells, of course, we would probably want to do a puromycin resistance. And that kind of goes into, this slide shows all the different things that are possible with ZFNs, including changing your promoters out or deleting and mutating them, making point mutations and gene corrections for disease models, new exons, reporter genes, and tags. You can also flox exons to do conditional knockouts if you wish, and we've made animals that way as well. And the different types of ZFN-related products are the custom ZFN, where you can basically pick the site and the area within that genetic locus that you want to either insert or mutate or flox. If you wanted to do a simple knockout, we have an off-the-shelf product, but the custom product is right here, where we design and assemble and validate in a relevant cell line. So everything that goes out the door is validated. Best performing ZFN is what you get either as plasmid only or plasmid plus mRNA. And it's about a 10 to 11 week delivery for the entire process. The knockout ZFNs are actually existing on our website as pre-designs. And these are validated in the same exact way. They're designed, assembled, and validated. The only difference is these are pre-designed for efficient knockout, not for any sort of knock-ins or conditionals or anything like that. Again, best performing ZFN is what we work up. And if the clone has already been validated, it's already banked, it takes 3 weeks to deliver. If not, we do the same pipeline and generate them in about 10 to 11 weeks. And we also have the targeted integration kits for human AAVS1 and mouse and rat ROSA 26 for integration into those loci, as I mentioned before. And the pricing for ZFNs has changed dramatically in the past 2 or 3 years. Basically, for the knockout ZFNs, DNA and DNA plus mRNA are at these 2 price points, about $4,000 and $5,000, and custom ZFNs are at $6,000 and $7,000. And the targeted integration kits, the safe harbor loci kits, are also very, very low priced. Now I'd like to do a brief overview of the transgenic animal creation that's been done with ZFNs. So this is just a diagram of the old way, the traditional way of creating knockout mice with stem cells. So you're using homologous recombination and a donor plasmid to make that genetic change in stem cells where the efficiency makes it possible. And then using those stem cells to inject into a blastocyst or a ball of cells to then create the organism that has the knockout. The downside of this is when you bring these into a ball of cells, there's always going to be some cells around that don't have the mutation. So you're not going to get germline immediately in many cases. You're going to have to backcross for months to get your animal to transmit that mutation into the offspring. So the difference is basically with ZFNs is now, because of the efficiencies of ZFNs, you can directly microinject them into the pronucleus. And this is of a single-celled embryo. Everything's going to go germline immediately because it's only one cell. So no mosaics, all the genetic changes go germline immediately, so you don't have to do all the backcrossing, and you don't need to do stem cell culture at all. You just need to microinject embryos. And this was proof of concept experiment in Science back in summer of 2009, where we targeted a GFP transgene in a GFP positive rat. So this is the other benefit. Now you don't have to just use mice. You can use rats or rabbits or anything else that you wish. And that GFP targeting ZFN was injected, and then we analyzed the pups. And for the first litter, you can see that instead of having the GFP phenotype, 2 of the pups now were GFP negative as the result of this 162 base pair deletion that was caused by the ZFN. And these were crossed with wild type and shown to go germline immediately as well. The same paper, they also did an endogenous gene, which was an IgM locus, and they analyzed those clones again with the PCR mismatch assay to look at some genomic DNA that was PCR'd at that locus, both wild type and mutated DNA. And because they're different, because the sequence is slightly altered, there are bubbles of non-homology, so they can cleave those with an enzyme called cell1 and run it on a gel. And when you run that on a gel, you'll see cleavage fragments that indicate there's some change in the genome of that organism. And then you can pick those clones and sequence to make sure that it's in fact an actual deletion or an insertion that will knock the gene out of frame. So Sigma was very inspired by this to go into possible exploration of diseases and other types of model organisms that are important for disease and biology. And what we did was we looked at areas that were sort of unmet in the field, especially of neurobiology, ADME-tox, cardiology, immunology, and cancer. And in many of these cases, there are much better phenotypes and much better models existing in the rat world. But historically, you couldn't do much with knockout rats. Now that we could, we decided to address that with a pipeline. This pipeline covers knockouts for genes that are important in various neurodegenerative diseases, such as Parkinson's, Alzheimer's. We also have pain models that are brand new, schizophrenia, autism. We just added those as well. ADME-tox, so drug efflux and transporters, immunology and oncology, and also cardiology. So these phenotypes are being characterized. Some of them have very, very well-established phenotypes where a mouse model didn't really confer such a great phenotype. So this allows people to do real work with biology and figure out these models of disease much more efficiently. On the flip side of that, if you don't see a gene that you want to use up here, you can actually create your own custom animal model knockout or knock-in. That's called the SAGE Speed service. And because of the efficiencies and the lack of backcrossing, we can do a knockout in about 5 to 6 months. we can do a knock-in in about 6 or 7 months, and about 8 or 9 months we can do humanizations and conditional knockouts. So this is all much faster than was ever before possible. And just to come full circle, you can see that we can do knock-ins into rat and mouse genes. This was just a small NOT1 insertion into a rat gene, and also into a mouse MDR1A, the same NOT1 site. Going a little bit larger, proof of concept, GFP cassette inserted into mouse MDR1A to come full circle and now restore that glowing green phenotype into our mouse, as evidenced here by the glowing green eyes. So we still have these available on special for Halloween. They're always favorites. So basically that's the talk, and that's the summary right there of the SAGE model creation. You can create these knockouts in about 5 to 7 months without ES cells and blastocysts, no backcrossing needed, and we can provide these as customs or as off-the-shelf models for you, and we can do the breeding as well. So that covers all of the ZFN talk. If anybody has any questions, I'll be glad to help out. Thank you so much.